440 million years ago, Earth was an alien world. No flowers painted meadows with color, no dinosaurs thundered across ancient plains, no grass carpeted the ground beneath your feet. The continents stretched bare and brown, dominated by stone and silence. Yet in this seemingly lifeless landscape, something extraordinary was happening, something that would change the very nature of our planet forever. Here's what was really occurring in those ancient times. The first fungi were learning to feed on darkness itself. Picture the Earth as it existed then. Imagine stepping onto those primordial shores where waves crashed against naked rock. The land before you would appear utterly barren, a moonscape of granite and sandstone baking under an alien sun. But look closer. Creeping across these sterile surfaces, almost invisible to the naked eye, were thin threads no wider than spider silk. These were the hyphae of the first terrestrial fungi, and they were performing an act of biological alchemy that had never been attempted before. These microscopic pioneers were doing something that seems impossible. They were eating stone. The universe had presented these early fungi with a problem that would challenge the very foundations of chemistry. How do you extract nutrition from solid rock? How do you break down granite, one of the hardest substances on Earth, using nothing but biological processes? The answer lay in a strategy so elegant, so revolutionary, that we're still marveling at its sophistication today. Fungi solved this puzzle by becoming living acid factories. Their thread-like hyphae began secreting organic acids powerful enough to dissolve the mineral bonds that had held granite together for millions of years. Imagine the scene: countless microscopic threads spreading across rock surfaces, each one releasing a carefully controlled mixture of citric acid, oxalic acid, and dozens of other compounds. These weren't random chemical attacks, but precisely targeted assaults on specific mineral components. The calcium feldspar in granite began to surrender its calcium ions. The quartz crystals started releasing their silicon. The mica sheets gave up their potassium and magnesium. For the first time in Earth's history, life was systematically dismantling the planet's rocky foundation and transforming it into something entirely new: soil. This process happened grain by grain, molecule by molecule. Each fungal thread would penetrate microscopic cracks in the rock, expanding them through sheer persistence and chemical warfare. The acids would flow into these crevices, working like molecular crowbars to pry apart mineral structures that had remained unchanged since the planet's formation. What emerged was a mixture of dissolved minerals, organic compounds, and loose rock particles, the very first soil in Earth's history. But here's where the story becomes even more remarkable. These fungi weren't working alone. Around the same time, perhaps within a few million years, with a blink of an eye, in geological terms, the first primitive plants were also making their tentative debut on land. These weren't the complex flowering plants we know today, but simple mosses and liverworts barely more sophisticated than their aquatic algae ancestors. They possessed the ability to photosynthesize, to capture sunlight and convert it into sugar, but they lacked the chemical weaponry to extract minerals from solid rock. The universe had created a perfect partnership waiting to happen. The fungi possessed the ability to dissolve stone and release essential minerals, but they couldn't manufacture their own food from sunlight. The plants could produce sugar through photosynthesis, but they couldn't access the nutrients locked away in granite and limestone. When these two groups discovered each other, they formed an alliance that would reshape the entire planet. This partnership, which scientists call mycorrhiza, literally means fungus root in Latin, but calling it a simple partnership understates its revolutionary nature. What developed was the world's first biological marketplace, where fungi traded dissolved minerals for plant-produced sugars in transactions so complex they mirror modern economic systems. Picture a primitive moss, barely visible against the rocky landscape, its simple leaves capturing precious photons from the ancient sun. Beneath the surface, fungal threads weave through the moss's rudimentary root system, creating a living network of chemical exchange. The moss pumps sugar molecules down to the fungi, while the fungi deliver a carefully measured package of dissolved minerals upward to the plant. The exchange rates fluctuated based on supply and demand, seasonal conditions, and the specific needs of both partners. But the fungi's ambitions extended far beyond simple trading partnerships. They had discovered something even more profound: the power of decomposition. In those early terrestrial ecosystems, death was beginning to accumulate. Plant matter that died had nowhere to go. Without decomposers, dead moss and primitive plant material would simply pile up, layer upon layer, creating vast organic graveyards. The carbon and nutrients locked in these dead tissues would become permanently unavailable to living organisms. Fungi looked at this accumulated death and saw opportunity. They developed the biochemical machinery to break down dead plant material, releasing the stored carbon and nutrients back into the ecosystem. But this wasn't simple digestion, it was a controlled demolition of organic molecules, conducted with surgical precision. The secret lay in their production of enzymes, protein machines capable of dismantling specific molecular structures. While other organisms could break down simple sugars and starches, fungi developed enzymes that could tackle cellulose, the tough structural polymer that gives plant cell walls their strength. This was biochemical pioneering work, requiring the evolution of entirely new classes of proteins. As millions of years passed, plants grew more sophisticated, and so did their partnership with fungi. Early tree-like plants began to appear, and with them came an even tougher challenge: lignin. Lignin is one of nature's most stubborn polymers. It's what makes wood woody, creating the rigid structural framework that allows trees to grow tall and strong. But lignin presented a problem that nearly broke the global carbon cycle. When these early trees died, their lignin-rich trunks and branches resisted decomposition. For 30 million years, dead wood accumulated across the planet's surface, creating the massive coal deposits we mine today. This was the Carboniferous period, literally the carbon-bearing age, when Earth nearly drowned in its own organic debris. The atmosphere became loaded with oxygen as plants photosynthesized, but their carbon remained locked in undecomposed wood. Oxygen levels soared to 35%, high enough to allow dragonflies with 2-foot wingspans to dominate the skies. Then, around 300 million years ago, fungi achieved their greatest biochemical breakthrough. They cracked the lignin code. Here's what's truly extraordinary about this achievement. Lignin is one of the most chemically resistant polymers in nature. Its molecular structure is so complex, so irregularly arranged, that it resists almost every form of biological and chemical attack. Yet fungi developed a suite of enzymes capable of systematically dismantling this molecular fortress. The key was white rot fungi, specialists that could produce lignin peroxidases and other oxidative enzymes. These molecular machines could break the critical bonds holding lignin together, reducing the toughest plant material to simple compounds that other organisms could then consume. For the first time since woody plants had evolved, dead trees could truly die, returning their stored carbon and nutrients to the living world. This breakthrough prevented our planet from becoming a world of organic debris. Without fungi's mastery of lignin degradation, Earth's surface would be buried under hundreds of millions of years of accumulated wood. The carbon cycle would have ground to a halt, and complex life as we know it could never have evolved. But fungi's early innovations extended beyond chemistry into architecture. During those ancient times, when the largest plants barely reached your knee, fungi were building structures that dwarfed everything else on land. Fossil evidence reveals fungal organisms 20 feet tall, rising from primordial soils like alien towers. These weren't mushrooms as we know them today, but massive fungal structures called prototaxites that dominated early terrestrial landscapes. Picture walking through forests where every tree was actually a giant fungus, their trunks composed entirely of densely packed fungal threads. These fungal giants raise questions that still puzzle scientists today. What evolutionary pressures created organisms capable of growing to such enormous sizes? How did they obtain enough nutrition to support such massive structures? And perhaps most mysteriously, what caused them to disappear, leaving behind only microscopic evidence of their former glory? The answers likely lie in the unique conditions of early Earth. With limited competition from plants and abundant organic matter accumulating on the surface, fungi found themselves in an ecological paradise. They could invest energy in growing upward, perhaps to better distribute their spores or to access different environmental conditions at varying heights. But as plants became more sophisticated and began competing more effectively for space and nutrients, these fungal giants found themselves evolutionary dead ends. The future belonged not to massive fungal towers, but to the intricate networks of microscopic threads that could weave through soil and plant roots with unprecedented efficiency. The legacy of these ancient innovations surrounds us today. Every grain of soil beneath your feet exists because fungi learned to dissolve stone 400 million years ago. Every forest depends on fungal partners to extract nutrients from the earth. Every decomposing leaf, every fallen tree, every organic molecule cycling through the biosphere does so because fungi mastered the chemistry of breaking down life's toughest polymers. When you walk through a forest today, you're walking through a world built by fungi, the soil that nourishes the trees, the nutrients that flow from root to leaf, the decomposing organic matter that feeds new growth. All of it depends on innovations, first developed by microscopic threads creeping across primordial stone. But perhaps most remarkably, only fungi mastered these essential biochemical processes. No other kingdom of life developed the ability to efficiently break down lignin. No other organisms became so sophisticated at dissolving minerals from solid rock. Fungi alone evolved the chemical arsenal necessary to connect the living and non-living worlds, to bridge the gap between biological and geological processes. This exclusivity raises profound questions about the nature of evolution and the role of fungi in Earth's history. Why didn't plants or animals develop similar capabilities? What unique aspects of fungal biology made them the planet's chosen decomposers and mineral extractors? The answers touch on fundamental principles of biochemistry, evolution, and the intricate relationships that make complex life possible. As we stand here today, 440 million years after fungi first learned to feed on darkness and stone, we're only beginning to appreciate the full scope of their achievements. These ancient innovators didn't just adapt to early Earth, they transformed it, creating the very foundations upon which all terrestrial life depends. The next time you see a mushroom pushing through forest soil, remember that you're looking at the descendant of organisms that taught Earth itself how to live. In learning to feed on darkness, fungi illuminated a path that led from barren stone to the rich, living world we inhabit today. Beneath your feet, right now, runs a network more complex than the human brain. As you stand reading these words, millions of microscopic threads are pulsing with information, trading resources, and coordinating the behavior of entire forests. This hidden internet connects every tree, every shrub, every blade of grass in an intricate web of communication that makes our most advanced computer networks look primitive by comparison. Here's what's actually happening in the soil beneath you. A single handful of forest earth contains over 100 miles of fungal threads, each one thinner than spider's silk, each one carrying messages that determine which plants live and which ones die. Scientists call this network the Wood Wide Web, and it operates on principles that challenge everything we thought we knew about plant behavior, forest ecology, and the nature of intelligence itself. To understand how extraordinary this system truly is, we need to journey into a world where trees are not the isolated individuals they appear to be, but connected participants in a vast underground society. Imagine you could shrink yourself down to the size of a sugar molecule and travel through this hidden realm. You would find yourself in a labyrinth of tunnels and chambers more intricate than any subway system ever built. Fungal hyphae, those microscopic threads we encountered in our first chapter, have evolved into something far more sophisticated than simple mineral extractors. They have become living cables, carrying information and resources across vast distances with precision that would impress any network engineer. Your journey would begin at a tree root, where you would witness one of nature's most elegant partnerships in action. The fungal threads don't just touch the root surface, they actually penetrate the root cells, creating hybrid structures where plant and fungus merge into something entirely new. These mycorrhizal connections are so intimate that it becomes impossible to say where the tree ends and the fungus begins. But here's where the story becomes truly remarkable. These connections aren't random. They're purposeful, directed, and they serve functions that read like science fiction. In 1997, a forest ecologist named Suzanne Simard made a discovery that revolutionized our understanding of forest ecology. Working in the forests of British Columbia, she designed an experiment that would reveal the secret life of trees. She injected different tree species with radioactive carbon isotopes, then used sensitive detectors to track where those carbon atoms traveled. What she discovered defied conventional wisdom about plant behavior. The carbon didn't stay in the trees where she had injected it. Instead, it moved through the forest, traveling from tree to tree through underground fungal networks. But the movement wasn't random, it was directed, purposeful, and it followed patterns that suggested something approaching intelligence. Mother trees, the largest and oldest trees in the forest, were actively nurturing their offspring. Through the fungal network, these ancient giants were sending precisely measured packages of carbon and nutrients to their struggling saplings, sometimes located hundreds of feet away in the deep shade of the forest floor. The mother trees could somehow identify their own genetic offspring and preferentially support them with resources, but the network's capabilities extended far beyond simple resource sharing. Simard's research revealed that trees were using the fungal internet to send chemical messages, warning their neighbors of impending dangers. Picture this scenario. Somewhere in the forest, an insect attack begins. Caterpillars start chewing on the leaves of a Douglas fir tree. Within minutes, the attacked tree begins flooding the fungal network with chemical alarm signals. These molecular messages race through the underground cables, traveling faster than the insects can spread. Trees hundreds of feet away receive these warnings and immediately begin producing defensive compounds, preparing for an attack that hasn't yet arrived. This communication system operates with a sophistication that challenges our understanding of plant intelligence. The chemical messages are specific, carrying detailed information about the type of threat, its severity, and even the precise defensive chemicals that are proving most effective. Receiving trees can tailor their responses based on this information, producing exactly the right cocktail of toxic compounds to defend against the specific insects heading their way. The speed of this communication network is astonishing. Chemical signals can travel through fungal hyphae at rates of up to 1 millimeter per second. That might sound slow compared to electrical signals in our nervous systems, but remember that we're talking about chemical information, traveling through living tissue over distances of hundreds of feet. A warning signal can reach every tree in a forest grove within hours of the initial attack. But the Wood Wide Web isn't just a communication network. It's also a sophisticated marketplace where resources are traded according to complex economic principles that would make Wall Street traders envious. The currency of this underground economy is simple. Trees trade sugar for minerals. Through photosynthesis, trees can manufacture all the sugar they need, but they struggle to extract essential nutrients like phosphorus, nitrogen, and potassium from the soil. Fungi excel at mining these nutrients from rock and soil, but cannot photosynthesize their own food. The result is a trading partnership that has evolved into something approaching a biological stock exchange. But here's what makes this marketplace truly extraordinary. The exchange rates fluctuate based on supply and demand, just like any modern economy. During summer months, when trees are photosynthesizing at peak capacity, sugar becomes relatively cheap in the underground markets. Trees can trade their abundant sugar for premium nutrients, supporting rapid growth and reproduction. But during winter, when photosynthesis slows or stops entirely, sugar becomes a precious commodity. Fungi adjust their trading rates accordingly, demanding more sugar for the same mineral packages. The complexity doesn't stop there. Different fungal species specialize in extracting different nutrients, creating a diverse marketplace where trees can shop for specific resources. Some fungi excel at extracting phosphorus from organic matter. Others specialize in mining potassium from granite. Still others focus on capturing nitrogen from the atmosphere or decomposing organic debris. Trees have learned to maintain relationships with multiple fungal partners simultaneously, diversifying their trading portfolios like savvy investors. A single tree might be connected to dozens of different fungal species, each offering different services at different rates. When one trading partner's prices become too high, the tree can shift its business to more competitive suppliers. This economic system includes quality control mechanisms that ensure fair trading. If a fungal partner begins delivering inferior nutrients or demanding excessive sugar payments, trees can reduce their investments in that relationship and seek better deals elsewhere. The fungi, dependent on the tree's sugar, must maintain competitive pricing and reliable service to retain their customers. Perhaps most remarkably, the network includes what economists would recognize as mutual aid societies and insurance systems during times of stress, drought, disease, or insect. Attack healthy trees will increase their support for struggling neighbors, even if those neighbors belong to different species and offer no immediate benefits in return. This cooperation violates basic economic theory, which predicts that organisms should always act in their own self-interest. Yet here we find trees using precious resources to help their competitors survive. The explanation lies in a principle that ecologists call the portfolio effect. Diverse forests are more stable and resilient than monocultures. By maintaining forest diversity, even at short-term cost to themselves, trees invest in the long-term stability of their entire ecosystem. The scale of these networks defies comprehension. In Oregon's Blue Mountains, researchers have identified a single organism, an Armillaria ostoyi fungus, that connects an entire forest across 3.4 square miles. This single organism, possibly the largest living thing on Earth, coordinates the activities of thousands of trees across an area larger than many cities. The Armillaria network pulses with electrical activity that bears striking resemblances to neural activity in animal brains. Using sensitive electrodes, researchers have detected rhythmic electrical signals traveling through the fungal network at regular intervals. These signals appear to coordinate the behavior of connected trees, synchronizing their growth patterns, defensive responses, and resource allocation decisions. The implications are staggering. We may be looking at a form of forest-scale intelligence, where individual trees function like neurons in a vast woody brain. The forest thinks, remembers, and makes collective decisions about resource allocation, defense strategies, and long-term planning. Each tree contributes its own processing power to a distributed intelligence that emerges from the connections between them. But the Wood Wide Web holds mysteries that challenge our deepest assumptions about intelligence and consciousness. How does the network store memory? How do fungal threads, lacking anything resembling a nervous system, process and route complex chemical information? How do mother trees identify their own offspring among the thousands of young trees in a forest? Recent research has revealed that the fungal networks can learn and adapt their behavior based on experience. Networks that have previously encountered particular threats respond more quickly and effectively when those threats return. The fungi appear to maintain some form of chemical memory, storing information about past events in molecular structures that can influence future behavior. Even more mysteriously, the networks demonstrate what appears to be decision-making behavior that transcends simple stimulus-response patterns. When resources are limited, the network somehow prioritizes which trees receive support, making complex calculations that consider each tree's age, health, genetic relationships, and strategic importance to the overall forest ecosystem. These decisions often favor outcomes that benefit the forest community over individual tree interests. Young trees in strategic locations might receive extra support to fill canopy gaps. Genetically diverse trees might be preferentially supported to maintain forest resilience. Old trees approaching death might have their resources redirected to support their offspring, ensuring genetic continuity across generations. The Wood Wide Web operates according to algorithms we cannot decode, processing vast amounts of environmental information to optimize forest health and productivity. It represents a form of collective intelligence that has been evolving for hundreds of millions of years, refining its strategies through countless generations of trial and error. As climate change and human development fragment forests worldwide, we're beginning to understand how crucial these underground networks are to forest survival. When we clear-cut forests or we build roads through wild areas, we're not just removing trees, we're severing communication cables and destroying information highways that took centuries to establish. Young forests planted on clear-cut land struggle to establish the complex underground networks that mature forests depend on. Without mother trees to provide guidance and support, without the accumulated wisdom stored in ancient fungal networks, these simplified forests remain vulnerable to drought, disease, and climate extremes. Scientists are now working to understand how we might restore these networks, how we might help damaged forests rebuild their underground internet. Some researchers are experimenting with inoculating tree seedlings with fungal partners before planting, giving young forests a head start in establishing their communication networks. Others are studying how to maintain network connectivity across human-disturbed landscapes, creating fungal bridges that allow forest fragments to share resources and information. The Wood Wide Web reminds us that the forest is not a collection of individual trees competing for resources, but a superorganism where cooperation and communication create emergent properties greater than the sum of their parts. Beneath our feet, a vast intelligence pulses with information and flows with resources, orchestrating the behavior of the living world in ways we are only beginning to comprehend. Every step you take through a forest is a step across living cables carrying messages between ancient minds. The trees around you are not silent, solitary beings, but participants in conversations that span continents and centuries. In the soil beneath your feet, the future of forests is being negotiated, one chemical message at a time. Deep in the rainforests of Thailand, something horrifying and precise is taking place. An ant worker, busy collecting food for her colony, suddenly stops mid-step. Her compound eyes, which moments before scanned the forest floor, with practiced efficiency, now stare fixedly upward. Her antennae, normally in constant motion sampling chemical cues, hang motionless. Something has invaded her mind. Here's what's actually happening to this ant. A microscopic fungal spore has breached her exoskeleton and is now flooding her nervous system with psychoactive compounds that will override 40 million years of evolved behavior. We are witnessing one of nature's most sophisticated acts of mind control, a biological puppet show that reveals fungi's terrifying mastery over consciousness itself. The ant begins to climb. This is not normal ant behavior. Worker ants should be foraging on the ground, not ascending into the canopy. But the fungus Ophiocordyceps unilateralis has hijacked her navigation system, reprogramming her most basic instincts with chemical precision. The infected ant climbs exactly 25 centimeters up the nearest plant stem. Not 20, not 30, but precisely 25 centimeters, the optimal height for fungal spore dispersal in these forest conditions. Upon reaching this predetermined height, the ant performs an action no healthy ant would ever consider. She turns 180 degrees to face north. Scientists still don't understand how the fungus accomplishes this navigational feat. The ant possesses no magnetic compass, no internal GPS system, yet the fungus somehow orients her body to face the precise direction that will maximize spore dispersal when winds blow from the south. Then comes the final, most disturbing act in this biological theatre. The ant clamps her mandibles onto the plant stem with 500 times her normal biting force. Her jaws lock so tightly that death itself cannot release them. She will remain attached to this stem long after the fungus consumes her from within, a biological anchor point for the fruiting body that will soon erupt from her head. But here's the most unsettling aspect of this entire process. The ant is still, alive during much of it. Brain scans of infected ants reveal that the fungus doesn't simply kill its host and animate the corpse. Instead, it keeps the ant's nervous system functional, while flooding it with mind-altering chemicals that rewrite the very essence of what it means to be an ant. The fungus produces compounds that mimic the ant's own neurotransmitters, hijacking communication pathways that evolution spent millions of years perfecting. But these aren't crude chemical sledgehammers that simply shut down brain function. They are precisely targeted molecular keys that unlock specific behavioral subroutines while leaving others intact. The infected ant retains her ability to walk, to climb, to grip with her mandibles. Her motor functions remain remarkably preserved, but her decision-making processes, her ability to respond to chemical signals from her colony, her instinct to return to the nest, all of these fundamental aspects of ant consciousness have been selectively disabled or reprogrammed. When researchers examine the brains of infected ants, they find something that challenges our understanding of how behavior is controlled at the cellular level. The fungus doesn't destroy brain tissue randomly. Instead, it appears to map the ant's neural networks, identifying specific circuits responsible for different behaviors, then systematically altering their function with surgical precision. The chemicals the fungus produces are so sophisticated that they can override instincts that have kept ant species alive for millions of years. The drive to return to the colony, one of the most fundamental behaviors in social insects, simply vanishes. The ant's alarm responses, her ability to recognize danger, her tendency to follow pheromone trails, all of these survival mechanisms are switched off as if someone had flipped a series of biological switches. But Ophiocordyceps represents just one example of fungi's mastery over consciousness. Across the globe, dozens of fungal species have evolved their own unique approaches to mind control, each one tailored to specific hosts and environmental conditions. In the Amazon, a different species of Ophiocordyceps infects bullet ants, the largest and most aggressive ants in the world. These formidable insects, whose sting is compared to being shot with a bullet, become docile passengers in their own bodies when infected. The fungus that hijacks bullet ants produces different chemicals than the one that controls smaller ant species, suggesting that each fungal puppet master has evolved a custom-designed neurochemical toolkit for its specific host. Some fungi take an even more dramatic approach to behavioral modification. Entomophthora muscae, which specializes in controlling flies, doesn't just change its host's behavior, it completely inverts it. House flies normally avoid elevated positions where they would be vulnerable to predators, but infected flies compulsively climb to the highest points they can find, spreading their wings wide and using their last moments of life to position themselves for optimal spore dispersal. The precision of these behavioral modifications suggests that fungi possess an understanding of animal nervous systems that rivals our own scientific knowledge. They have somehow decoded the chemical languages that neurons use to communicate, learning to speak fluent nerve in dozens of different dialects. But fungi's influence on consciousness extends far beyond mere mind control. Thousands of miles from tropical rainforests, in the laboratories of Johns Hopkins University and other research institutions, Scientists are studying how certain fungi produce compounds that don't hijack consciousness, but expand it in ways that challenge our understanding of the mind itself. Psilocybe mushrooms, commonly known as magic mushrooms, produce a compound called psilocybin that bears striking resemblances to serotonin, one of the most important neurotransmitters in the mushroom. Human brain. This molecular similarity allows psilocybin to bind to the same receptors that serotonin uses, but with profoundly different effects. When psilocybin enters the human brain, it doesn't simply mimic serotonin's normal functions. Instead, it appears to dissolve the barriers between different brain regions, allowing areas that normally operate independently to communicate directly with each other. Brain imaging studies reveal that people under the influence of psilocybin show dramatically increased connectivity between brain regions, creating neural communication patterns that never occur in ordinary consciousness. The results are experiences that participants describe as among the most meaningful in their lives. People report profound insights into the nature of reality, feelings of unity with the universe, and therapeutic breakthroughs that conventional treatments couldn't achieve. Some participants describe these experiences as more real than ordinary reality, suggesting that psilocybin doesn't distort consciousness, but rather reveals aspects of it that are normally hidden. Here's what makes this phenomenon so remarkable. Fungi evolved psilocybin millions of years before humans existed. These mushrooms didn't develop psychoactive compounds to interact with human consciousness. They evolved them for their own purposes, possibly to deter insects or communicate with other fungi. Yet the compounds they produce unlock doors in human awareness with a precision that seems almost intentional. The anthropologist Terence McKenna proposed a controversial theory that fungi may have catalyzed human language evolution. He suggested that early humans who consumed psilocybin mushrooms experienced enhanced pattern recognition and communication abilities that accelerated the development of complex language. While this theory remains speculative, research is revealing that psilocybin does indeed enhance creativity, empathy, and novel thinking patterns in ways that could theoretically boost cognitive evolution. But fungi's mastery over consciousness extends beyond chemical manipulation. Some species demonstrate forms of intelligence that challenge our most basic assumptions about what consciousness requires. Slime molds like Physarum polycephalum accomplish feats of problem-solving that would impress graduate students, despite lacking anything resembling a nervous system. These single-celled organisms can navigate complex mazes, finding the shortest path between multiple food sources with mathematical precision. When researchers placed slime molds in replicas of Tokyo's transportation network, the organisms spontaneously recreated the city's rail system, building connections between major hubs that matched the efficiency of human-designed infrastructure. The slime mold achieves these remarkable feats through a form of distributed intelligence that operates on completely different principles from brain-based consciousness. As the organism explores its environment, it leaves behind chemical trails that mark successful and unsuccessful paths. Over time, these chemical memories create a map of the most efficient routes through complex territories. But here's what's truly extraordinary. The slime mold doesn't just follow these chemical maps passively, it continuously updates and optimizes them based on changing conditions. When food sources move or disappear, the organism rapidly reorganizes its network, finding new solutions that often surpass the efficiency of the previous arrangement. This suggests a form of learning and memory that operates without neurons, without brains, without any of the biological structures we associate with intelligence. The slime mold stores information in chemical gradients, processes it through cellular mechanisms, and makes decisions that demonstrate planning, optimization, and adaptation. The implications are staggering. If a single-celled organism can demonstrate these cognitive abilities, what might more complex fungal networks be capable of? The mycorrhizal networks we explored in our previous chapter suddenly take on new significance. If individual fungi can solve mazes and optimize transportation networks, What kinds of problems might forest-scale fungal networks be tackling? Researchers studying these questions have documented behaviors that suggest fungal networks possess forms of awareness that transcend our understanding of consciousness. The mycologist Paul Stamets has reported that mycelium cultures respond differently to different human visitors, growing towards some people while retreating from others. These responses appear to be consistent and repeatable, suggesting that the fungi can somehow distinguish between individual humans. Other researchers have documented fungal networks that seem to anticipate future events, adjusting their growth patterns and resource allocation in advance of environmental changes that haven't yet occurred. How they accomplish this apparent precognition remains a complete mystery. The study of fungal consciousness forces us to confront fundamental questions about the nature of mind itself. We've long assumed that consciousness requires a centralized nervous system, that intelligence emerges from the complex interactions of billions of neurons. But fungi demonstrate that awareness can arise from completely different biological architectures. Their intelligence appears to be distributed, collective, and fundamentally alien to human experience. Where we have individual minds housed in discrete bodies, fungi appear to possess collective consciousness that spans vast networks of interconnected organisms. Where we think in terms of discrete thoughts and linear reasoning, fungi seem to process information holistically, through chemical gradients and network dynamics that we're only beginning to understand. Perhaps most unsettling of all, fungi's mastery over consciousness suggests that the boundaries between self and other, between one mind and another, may be far more fluid than we imagine. When Ophiocordyceps takes control of an ant, where does the ant end and the fungus begin? When psilocybin dissolves the barriers between brain regions, are we experiencing fungal consciousness merging temporarily with human awareness? These questions don't have easy answers, but they point toward a truth that challenges our most basic assumptions about the nature of mind and consciousness. We share this planet with forms of intelligence so alien, yet so sophisticated, that understanding them might reveal new dimensions of what it means to be aware, to think, to experience the universe. As you read these words, millions of fungal networks are processing information, making decisions, and perhaps even experiencing forms of consciousness that our language lacks words to describe. The boundary between mind and matter, between intelligence and chemistry, between self and other, may be far more permeable than we ever imagined. Inside each mushroom cell, chemical factories are operating that shouldn't exist according to everything we understand about biochemistry. These microscopic laboratories produce compounds that defeat cancer, devour petroleum, digest radioactive materials, and perform chemical transformations that our most advanced technology cannot replicate. We are about to journey into a realm where fungi routinely violate the laws of chemistry as we know them. Here's what's actually happening inside these cellular factories. Fungi are accomplishing chemical reactions at room temperature and atmospheric pressure that our industries can only achieve using extreme heat, crushing pressure, and toxic solvents. They are nature's master chemists, wielding molecular tools that make our pharmaceutical laboratories look primitive by comparison. To understand just how extraordinary fungal chemistry truly is, we need to travel to one of the most dangerous places on Earth, the ruins of Chernobyl's Reactor 4. On April 26, 1986, Reactor 4 at the Chernobyl nuclear power plant exploded, releasing radioactive material equivalent to 400 times the atomic bomb dropped on Hiroshima. The surrounding area became so contaminated that it was declared uninhabitable for the next 20,000 years. Nothing should be able to survive in the reactor's core, where gamma radiation levels remain lethal to most life forms. Yet when scientists finally gained access to the reactor's interior, they made a discovery that defied every principle of biology they thought they understood. Growing on the reactor walls, thriving in radiation levels that would kill a human in minutes, they found thick black mats of fungal growth. The fungus Cryptococcus neoformans wasn't just surviving in this radioactive hellscape, it was flourishing. Here's what makes this discovery so revolutionary. These fungi appear to be using radiation as an energy source, converting deadly gamma rays into food through a process that redefines our understanding of how life can sustain itself. The secret lies in melanin, the same pigment that gives color to human skin and hair. In these radiation-eating fungi, melanin serves as a biological solar panel, capturing high-energy gamma rays and converting them into chemical energy through a process that bears striking resemblances to photosynthesis. But while photosynthesis uses visible light, these fungi have learned to harvest the most dangerous forms of electromagnetic radiation. The biochemical mechanisms involved challenge everything we thought we knew about the limits of life. Gamma radiation typically destroys biological molecules by ripping electrons away from atoms, creating highly reactive free radicals that tear apart cellular machinery. But these fungi have evolved melanin structures that can capture those same high-energy electrons and channel them into productive biochemical pathways. It's as if these organisms have transformed radiation, one of life's greatest enemies, into their primary food source. The implications are staggering. If fungi can eat radiation, what other supposedly toxic or dangerous compounds might they be capable of consuming? The answer has emerged from laboratories and contaminated sites around the world, where researchers have discovered fungi capable of digesting some of the most persistent pollutants on the planet. Pleurotus ostreatus, commonly known as the oyster mushroom, possesses the ability to break down petroleum products with an efficiency that surpasses our best environmental cleanup technologies. When this fungus encounters diesel fuel, motor oil, or other petroleum contamination, it produces a suite of enzymes that systematically dismantle these complex hydrocarbons, molecule by molecule. The process begins when the fungal hyphae detect petroleum compounds in their environment. Within hours, they begin secreting lignin peroxidases, the same enzymes we learned about in our first chapter that break down wood. But here's the remarkable part. These enzymes don't just break down lignin. They can also cleave the carbon-carbon bonds in petroleum molecules, reducing toxic hydrocarbons to harmless carbon dioxide and water. In controlled experiments, oyster mushroom cultures have transformed diesel-contaminated soil into fertile earth in just 90 days. The fungi don't simply sequester the pollutants, they actually digest them, converting environmental toxins into mushroom biomass and soil nutrients. Where contaminated land once supported no life, mushroom-treated sites become lush with new plant growth. But fungi's chemical capabilities extend far beyond breaking down pollutants. They are also master synthesizers, capable of producing pharmaceutical compounds with precisions that rival our most sophisticated drug manufacturing facilities. The story begins with a chance observation in 1928, when Alexander Fleming noticed that a mold growing in his bacterial cultures had created a clear zone where no bacteria could survive. That mold, Penicillium notatum, was producing a compound that would become penicillin, the world's first antibiotic. But Fleming's discovery was just the beginning. The penicillin molecule represents just one example of fungi's vast pharmaceutical arsenal. These organisms produce thousands of bioactive compounds, many of which remain completely unknown to science. Each species appears to maintain its own unique chemical library, evolved over millions of years of biological warfare with bacteria, viruses, and competing fungi. The sophistication of these natural drug factories becomes apparent when we examine how they work. Fungi don't just randomly produce toxic compounds and hope for the best. They manufacture precise molecular keys designed to target specific biological processes in their competitors or predators. Take the ergot alkaloids produced by Claviceps purpurea, a fungus that grows on rye and other grasses. These compounds mimic neurotransmitters in the human nervous system with such precision that they can trigger hallucinations, convulsions, and death in tiny doses. But they're not random poisons. They're carefully crafted molecules that bind to specific receptor sites in nerve cells, disrupting neural communication with surgical precision. The pharmaceutical industry has learned to harness these fungal capabilities, using ergot alkaloids to treat migraines, control bleeding during childbirth, and even explore consciousness through compounds like LSD, which is derived from ergot chemistry. But we're only scratching the surface of fungi's pharmaceutical potential. Modern drug discovery programs routinely screen fungal extracts for new therapeutic compounds, and they continue to find molecular treasures that boggle the mind. Fungi produce natural antibiotics that target drug-resistant bacteria, anti-cancer compounds that selectively destroy tumor cells while leaving healthy tissue untouched, and immunomodulatory drugs that can boost or suppress immune responses with remarkable specificity. One of the most mysterious examples of fungal pharmaceutical prowess comes from turkey tail mushrooms, whose scientific name, Trametes versicolor, hardly conveys their remarkable healing properties. These common bracket fungi produce polysaccharides that boost human immune function through mechanisms that seem almost impossibly sophisticated. Here's what makes turkey tail's immune-boosting effects so puzzling. The polysaccharides these fungi produce appear to be perfectly designed to interact with human immune system receptors. The molecular structures match our cellular recognition systems so precisely that they could have been custom designed in a laboratory to optimize human health. But turkey tail mushrooms evolved these compounds millions of years before humans existed. How could natural selection have shaped molecules to interact so perfectly with immune systems that wouldn't evolve for eons? It's as if fungi anticipated the future evolution of mammalian immunity and prepared healing compounds in advance. This phenomenon appears again and again in fungal chemistry. Compounds that seem tailor-made to interact with human biology, produced by organisms that had no evolutionary contact with our species. Reishi mushrooms produce triterpenes that calm human anxiety through mechanisms that target our stress response systems. Cordyceps fungi produce compounds that enhance human athletic performance by optimizing oxygen utilization in our cells. The precision of these interactions suggests that fungi have decoded fundamental principles of biochemistry that we're only beginning to understand. They appear to recognize molecular patterns and cellular processes that are conserved across vast evolutionary distances, allowing them to produce compounds that work across multiple species despite having no direct evolutionary relationship with their targets. But perhaps the most mind-bending aspect of fungal chemistry is their ability to perform chemical transformations that we cannot replicate in our laboratories, despite decades of research and billions of dollars in investment. Consider the biosynthesis of penicillin, which requires the formation of a highly strained four-membered ring structure called a beta-lactam. This molecular arrangement is so unstable that it requires extraordinary chemical finesse to create. Synthetic chemists can now manufacture penicillin industrially, but only through complex multi-step processes that require harsh chemicals, extreme temperatures, and precisely controlled conditions. The penicillium fungus accomplishes the same chemical transformation at room temperature, using nothing but enzymes and the chemical building blocks available in ordinary organic matter. The fungal assembly line that produces penicillin operates with such efficiency that it puts our best pharmaceutical manufacturing to shame. This pattern repeats across the entire spectrum of fungal chemistry. Complex molecular structures that require our most advanced synthetic methods are produced routinely by fungi using simple, environmentally friendly processes. They accomplish in single steps what takes our chemists dozens of reactions to achieve. The enzymes that catalyze these reactions represent biological machines of almost incomprehensible sophistication. Each enzyme is a protein molecule folded into a precise three-dimensional shape that creates a molecular workspace where specific chemical transformations can occur. The active sites of these enzymes position reactant molecules with atomic-level precision, facilitating reactions that would be virtually impossible in ordinary chemical conditions. But fungi don't just produce individual enzymes, they orchestrate entire enzymatic assembly lines that carry out complex synthetic pathways with remarkable coordination. Raw materials enter at one end of the pathway and emerge as finished pharmaceutical compounds at the other, with each step precisely timed and controlled to optimize efficiency and minimize waste. The regulatory systems that control these pathways demonstrate a level of chemical intelligence that we're only beginning to appreciate. Fungi can sense environmental conditions and adjust their chemical production accordingly, ramping up antibiotic synthesis when bacterial threats are detected, or switching to different metabolic pathways when nutrients become scarce. They can even modify their chemical output in real time based on the responses of their targets. If bacteria begin developing resistance to one antifungal compound, the fungus can shift production to alternative chemicals that exploit different vulnerabilities. It's biological chemical warfare, conducted with a strategic sophistication that rivals human military planning. As we face challenges like antibiotic resistance, environmental pollution, and the need for sustainable manufacturing processes, fungi offer us a glimpse of what truly advanced chemistry looks like. These organisms have spent hundreds of millions of years perfecting chemical processes that we're only beginning to understand. They remind us that the most sophisticated chemistry laboratory on Earth isn't built by humans, it's grown by fungi, one microscopic cell at a time. In their impossible chemistry, we find both humility about our current limitations and inspiration for what biotechnology might eventually achieve. In the pitch-black depths of New Zealand's Waitomo caves, something impossible illuminates the darkness. Thousands of tiny blue lights speckle the cave ceiling like an underground galaxy, creating a constellation so beautiful that tourists travel across the world to witness this natural wonder. But here's what makes this bioluminescent display truly extraordinary. It shouldn't exist according to everything we understand about energy conservation in living systems. The lights come from Arachnocampa luminosa, glowworm larvae, whose fungal partners have somehow mastered the art of cold light production. These organisms generate a blue radiance pure enough to read by, using luciferin reactions that consume almost no energy and produce virtually no waste heat. While our most efficient LED lights convert only 20% of their energy into visible light, these fungal-animal partnerships achieve efficiencies approaching 100%. Here's what's actually happening in these living light bulbs. The fungal partners have evolved biochemical processes that challenge the fundamental laws of thermodynamics as we understand them. They produce light through chemical reactions that operate at near-theoretical efficiency levels, creating photons with almost no energy loss to heat or chemical byproducts. The mechanism involves luciferin, a light-producing compound, and luciferase, an enzyme that catalyzes its oxidation. But the fungal version of this chemistry operates with a precision that surpasses anything we can achieve artificially. The enzyme-substrate interactions are so perfectly optimized that virtually every chemical reaction produces a photon, with no energy wasted on unproductive side reactions. But bioluminescence represents just one of many fungal phenomena that transcend our understanding of biological possibility. Across the planet, fungi routinely demonstrate capabilities that read like violations of natural law. In grasslands from Ireland to Australia, mysterious circles appear overnight, growing steadily outward at exactly 13 inches per year. These fairy rings, as folklore has named them, create geometric patterns so precise they can be observed from space satellites, some expanding in perfect circles for over 700 years. The rings form when underground fungal networks exhaust nutrients in their center and spread outward in search of fresh resources. But here's what defies explanation: the mathematical precision of their expansion. Despite variations in soil composition, moisture levels, seasonal weather patterns, and countless other environmental variables, These rings maintain their 13-inch annual growth rate with clockwork consistency. How do the fungi measure distance with such accuracy? How do they coordinate their growth to maintain perfect circular geometry across terrain that varies dramatically in elevation, soil chemistry, and vegetation? The mechanisms behind this precision remain completely unknown, yet the phenomenon persists across dozens of fungal species and every continent where grasses grow. Some fairy rings predate Stonehenge, expanding outward for millennia with unwavering mathematical precision. These ancient circles represent living geometric theorems, biological compasses that trace perfect curves across landscapes with an accuracy that would challenge our most sophisticated surveying equipment. But fairy rings pale in comparison to the ballistic capabilities displayed by Pilobolus crystallinus, a fungus that has evolved into nature's most sophisticated artillery system. This tiny organism, commonly found growing on herbivore dung, faces a precise targeting challenge. It must launch its spores far enough to escape the toxic environment where it grows, but accurately enough to land on fresh grass that animals will eat, continuing its life cycle. The solution Pilobolus has evolved reads like engineering fiction. Each fruiting structure functions as a microscopic cannon, complete with a focusing lens, pressure chamber, and targeting system. The organism literally grows its own optics, creating a transparent vesicle that focuses sunlight with the precision of a telescope lens. Here's where the physics becomes truly extraordinary. Pilobolus uses the focused sunlight to heat a pressurized chamber filled with cellular fluid. As pressure builds, the organism aims its biological cannon by bending toward the brightest light source, automatically targeting areas where fresh grass is most likely to be growing. When pressure reaches critical levels, the spore package launches with an acceleration that exceeds 65 feet per second, the fastest acceleration recorded in the natural world. But the targeting system demonstrates capabilities that challenge our understanding of fungal sensory abilities. The organism can distinguish between direct sunlight and reflected light, preferentially aiming toward areas that receive the most direct illumination. This suggests a level of optical sophistication that rivals the compound eyes of insects, achieved by an organism that has no recognizable visual system. The ballistic calculations involved in each shot would challenge a military engineer. The fungus must account for wind resistance, gravitational drop, and the optimal trajectory to achieve maximum distance while maintaining accuracy. Yet it accomplishes these computations using nothing but cellular machinery and simple chemical gradients. Even more mysteriously, Pilobolus demonstrates what appears to be learning behavior. Colonies that have successfully launched spores toward productive targets seem to improve their accuracy over time, suggesting some form of feedback mechanism that allows them to refine their targeting algorithms based on previous results. But perhaps the most perplexing of all fungal phenomena is synchronized fruiting, where millions of mushrooms across vast geographic distances emerge within hours of each other, responding to signals that remain completely undetectable to our instruments. The phenomenon occurs repeatedly across multiple species and continents. After weeks or months of underground growth, mushroom colonies spanning hundreds of miles will simultaneously push their fruiting bodies to the surface, creating overnight mushroom blooms that can cover entire regions. The synchronization is so precise that mycologists can predict these events to within a few hours, yet the triggering mechanisms remain completely unknown. Weather patterns don't explain the synchronization. Mushroom emergences often occur during stable weather conditions, with no obvious environmental triggers. Seasonal cues fail to account for the precision of the timing, as synchronized fruiting can happen at any time of year when conditions are generally favorable. Some researchers have proposed that fungi respond to subtle electromagnetic fields, perhaps generated by solar activity or geomagnetic fluctuations. Others suggest that chemical signals might travel through underground fungal networks, coordinating fruiting across vast distances. But no detection equipment has ever identified the signals responsible for these coordinated events. The precision of synchronized fruiting suggests communication systems that operate beyond our current understanding of biological signaling. How do fungal networks across different continents coordinate their activities without any apparent means of long-distance communication? What environmental cues are they detecting that our most sensitive instruments cannot measure? Adding to the mystery are reports from researchers worldwide of mushroom circles that affect electronic equipment in ways that science cannot explain. Compass readings become erratic when taken within certain fairy rings. Digital cameras experience electronic interference near some mushroom colonies. Temperature sensors register anomalous readings that suggest localized energy fields around fungal fruiting bodies. Infrared cameras have captured thermal anomalies around some mushroom colonies, showing temperature variations that don't correspond to any known biological processes. These warm and cool zones form geometric patterns that mirror the underground fungal networks, suggesting that the organisms might be generating or manipulating energy fields in ways we cannot detect or understand. Time-lapse photography has revealed even stranger phenomena. Footage from forest monitoring stations occasionally captures mushroom networks that appear to pulse with coordinated rhythms, their fruiting bodies expanding and contracting in synchronized waves that span multiple acres. The pulsing patterns don't correspond to day-night cycles, weather changes, or any other environmental variables that researchers have been able to identify. Most intriguingly, some of these pulsing rhythms appear to match astronomical cycles with uncanny precision. Researchers have documented mushroom networks whose expansion patterns correlate with lunar phases, solar magnetic field fluctuations, and even the gravitational effects of planetary alignments. The correlations are too precise to be coincidental, yet the mechanisms behind them remain completely unknown. These observations have led some scientists to propose that fungi might be sensitive to gravitational waves, quantum field fluctuations, or other fundamental forces that we're only beginning to understand. The idea sounds like science fiction, yet the data continues to accumulate, documenting fungal behaviors that transcend conventional biological explanations. In Japanese forests, researchers have recorded mushroom colonies that seem to respond to human presence in ways that suggest awareness extending beyond simple chemical detection. Some fungi increase their growth rates when researchers approach, while others retreat or cease activity entirely. The responses appear to be species-specific and consistent across multiple observation sessions, suggesting that different fungi have developed different strategies for dealing with human contact. Even more puzzling are reports of mushroom colonies that appear to anticipate human visits, beginning to change their behavior hours or even days before researchers arrive at study sites. These anticipatory responses cannot be explained by chemical cues, vibrations, or any other conventional sensory mechanisms. The accumulated evidence points toward fungal capabilities that challenge our most basic assumptions about the limits of biological sensory systems. These organisms appear to be detecting and responding to environmental signals that exist beyond the range of human perception and scientific instrumentation. Perhaps most remarkably, some researchers have begun to document what appears to be fungal memory extending across multiple fruiting cycles. Mushroom colonies that experience particular environmental challenges during one growing season appear to remember those experiences and modify their behavior during subsequent seasons, even when the original stressors are no longer present. This suggests that fungal networks might be maintaining information storage systems that persist across multiple generations of fruiting bodies. The implications are staggering. If fungi can store and retrieve experiential information across decades or centuries, they might possess historical records of environmental changes that extend far beyond human written history. As our instruments become more sensitive and our understanding of physics expands into quantum realms and gravitational wave detection, we may discover that fungi have been responding to fundamental forces that we're only beginning to recognize. These ancient organisms might be natural sensors for phenomena that exist at the very edges of scientific knowledge. The fungal kingdom presents us with a catalogue of impossible phenomena that challenge the boundaries between biology and physics, between the possible and the inexplicable. In their mysterious capabilities, we find reminders that the natural world operates according to principles that extend far beyond our current understanding, governed by laws we have yet to discover. In a factory in California, something that sounds like science fiction is becoming everyday reality. Designers feed sugar water to colonies of mushroom mycelium, and 14 days later, they harvest sheets of material that look, feel, and perform like leather. No animals are harmed, no toxic chemicals are used, and no years of waiting are required. This is the dawn of the fungal revolution, where humanity finally learns to harness the extraordinary capabilities we've been exploring throughout our journey. Here's what's actually happening in these biomanufacturing facilities. Companies are training fungi to grow into predetermined shapes and textures, creating materials that match or exceed the performance of traditional products, while using a fraction of the resources and producing zero toxic waste. The process begins with mycelium, those microscopic fungal threads that we've encountered in every chapter of our story. But instead of letting them grow wild underground, engineers are learning to guide their development with the precision of software programmers writing code. By controlling nutrients, moisture, temperature, and even mechanical stress, they can direct fungal growth to create materials with specific properties. The mycelium responds by weaving itself into dense, fibrous mats that can be processed into leather alternatives, packaging materials, building insulation, and even textiles. The fungi essentially grow the product to specification, creating complex three-dimensional structures that would be impossible to manufacture using traditional methods. But this is just the beginning. The same companies are now experimenting with programming fungi to grow into architectural components, living building materials that strengthen over time rather than deteriorating. Imagine walking into a house where the walls are alive. The insulation isn't made from petroleum-based foam or mineral fibers, but from living mycelium networks that actively regulate temperature, humidity, and air quality. These fungal walls breathe with the seasons, expanding their insulating properties during winter, increasing ventilation during summer months. When damaged, they don't need to be replaced, they heal themselves. A crack in a mycelium wall triggers the fungal network to increase growth in that area, literally growing new material to fill the gap. The repair happens automatically, without human intervention, using nutrients the fungi extract from the air and moisture. The air purification capabilities of these living buildings surpass anything current technology can achieve. The fungal networks actively filter toxins, allergens, and pollutants from indoor air, converting harmful compounds into harmless byproducts. They can even be programmed to target specific contaminants, creating buildings that adapt their air filtering to local environmental conditions. But architects are dreaming even bigger. Research teams are developing fungal networks that could form the structural framework of entire buildings, creating self-assembling architecture that grows from seed to completion without traditional construction methods. The process would begin with inoculating a building site with specially designed fungal spores and a network of nutrients embedded in biodegradable scaffolding. The fungi would grow according to architectural plans encoded in their genetic programming, forming walls, support beams, and even complex interior features as they develop. These bioarchitectural structures would be stronger than traditional buildings because the fungal networks would form continuous, interconnected webs rather than separate components bolted together. They would be self-repairing, environmentally responsive, and capable of adapting their properties as needs change over decades of use. Meanwhile, in space agencies around the world, fungi are being prepared for humanity's next great adventure, the colonization of other planets. The challenges of space colonization have always centered on the prohibitive cost and complexity of transporting everything humans need to survive. Every pound of material launched to Mars costs thousands of dollars, making traditional colonization strategies economically impossible. But fungi offer a revolutionary alternative. Instead of shipping finished products to space, we can ship fungal spores and let them manufacture everything we need on arrival. Experiments aboard the International Space Station have already demonstrated that fungi can process human waste into food, oxygen, and building materials in the closed-loop life support systems that space colonies will require. The fungi consume organic waste that would otherwise accumulate and become toxic, converting it into edible protein, breathable oxygen, and structural materials for expanding habitation modules. But the applications extend far beyond waste processing. Fungal networks could terraform entire planetary surfaces, breaking down alien soil compositions and establishing the biological foundations necessary for human agriculture. They could process atmospheric gases on planets like Mars, potentially extracting water from the atmosphere and soil while generating oxygen as a byproduct. Some researchers envision fungal networks serving as the primary life support infrastructure for space colonies, creating self-sustaining biological systems that maintain atmospheric composition, process waste, produce food, and even generate building materials from local planetary resources. Back on Earth, the medical applications of fungal biotechnology are revolutionizing our approach to mental health treatment, with results that seemed impossible just a decade ago. Clinical trials using psilocybin, the psychoactive compound from magic mushrooms, are achieving therapeutic breakthroughs that conventional treatments couldn't accomplish in decades of effort. Patients with treatment-resistant depression, post-traumatic stress disorder, and addiction are experiencing lasting recovery after just 1 or 2 guided psilocybin sessions. Here's what makes these results so extraordinary. Brain imaging studies show that psilocybin literally rewires depressed brains, creating new neural connections that persist long after the immediate effects of the compound have worn off. The treatment appears to break patients out of negative thought patterns by temporarily dissolving the brain networks that maintain depressive thinking, allowing new, healthier neural pathways to form. The implications extend far beyond treating existing mental health conditions. Researchers are exploring whether psilocybin could enhance creativity, empathy, and problem-solving abilities in healthy individuals, potentially augmenting human cognitive capabilities in ways that could accelerate scientific discovery and artistic innovation. But perhaps the most revolutionary application of fungal biotechnology lies in their potential to reverse 2 centuries of environmental damage by sequestering atmospheric carbon at industrial scales. Fungi naturally store carbon in soil through their vast underground networks, converting atmospheric CO2 into stable organic compounds that can remain sequestered for decades or centuries. But researchers are now engineering fungal systems specifically designed to maximize carbon capture and storage. These enhanced fungi can sequester 17 tons of carbon dioxide per acre, while simultaneously regenerating degraded soil, restoring fertility to agricultural land that has been depleted by industrial farming practices. The process is completely natural and self-sustaining. The fungi require no external energy inputs beyond what they extract from sunlight and atmospheric gases. Large-scale deployment of carbon-sequestering fungi could theoretically reverse the atmospheric effects of the entire industrial revolution within decades, while simultaneously restoring millions of acres of degraded farmland to productivity. The fungi essentially mine carbon from the atmosphere and deposit it in soil where it supports new plant growth and ecosystem recovery. But the most intriguing possibility emerging from fungal biotechnology research is the development of biological computing systems that process information through chemical signals rather than electrical circuits. Fungal networks naturally process vast amounts of environmental information, making complex decisions about resource allocation, threat response, and growth patterns through chemical communication systems we're only beginning to understand. Researchers are now learning to interface with these biological information processing networks, essentially creating living computers that think in chemistry rather than electricity. These fungal computers could theoretically achieve forms of consciousness that are alien to human experience but equal in complexity and sophistication. They might process information holistically rather than sequentially, think in chemical gradients rather than digital states, and solve problems through biological algorithms that evolved over hundreds of millions of years. The possibility of communicating with such systems raises profound questions about the nature of intelligence and consciousness. If we can learn to speak the chemical languages that fungi use to process information, we might establish communication with Earth's oldest and most widespread intelligence network. Such communication could revolutionize our understanding of ecology, consciousness, and our place in the biosphere. Fungal networks possess environmental information spanning centuries or millennia, encoded in chemical memories that could teach us about climate patterns, ecosystem dynamics, and biological relationships that predate human civilization. As these technologies mature, they're converging toward a future where the boundary between biology and technology disappears entirely. Buildings will be grown rather than constructed. Materials will heal themselves when damaged. Computers will think in chemistry rather than electricity. Space colonies will be ecosystems rather than machines. The fungal revolution represents more than just new technologies. It's a fundamental shift toward working with biological systems rather than against them, toward harnessing evolution solutions rather than imposing our own crude alternatives. Fungi spent 400 million years perfecting the technologies we're only now learning to appreciate. They mastered materials science before materials existed, developed information processing systems before brains evolved, and created global communication networks before nervous systems were invented. As we face challenges like climate change, resource depletion, and the need for sustainable technology, fungi offer us proven solutions that have been tested across geological timescales. Their technologies are self-repairing, environmentally responsive, and capable of adapting to changing conditions without human intervention. The companies and researchers leading this revolution aren't just developing new products, they're pioneering an entirely new relationship between humanity and the natural world. Instead of extracting resources from nature, they're learning to partner with biological systems that offer capabilities we could never achieve through purely mechanical approaches. In this fungal future, the question isn't whether we'll survive our environmental challenges, but whether we're wise enough to accept the partnership that fungi have been offering us for millions of years. The invitation has always been there, written in chemical languages we're only now learning to read, waiting for us to recognize that the future isn't something we need to build, it's something we need to grow. After centuries of study, after mapping their genomes and cataloging their chemistry, after witnessing their impossible phenomena and harnessing their revolutionary technologies, fungi continue to guard secrets that humble human knowledge. We stand at the edge of understanding, peering into mysteries that challenge our most fundamental assumptions about life, intelligence, and consciousness itself. Here's what we still don't understand: why some mushroom species wait precisely 70 years to fruit, responding to celestial events that our instruments cannot detect. Why fungi without any DNA coding for photosynthesis can capture and utilize light in ways that defy the laws of physics as we know them. Why over 80 species invest precious energy in bioluminescence when the survival advantages remain completely unclear. These questions aren't merely academic curiosities. They point toward capabilities and awareness that suggest fungi operate according to principles we haven't discovered yet. Consider the 70-year fruiting cycles observed in certain bamboo-associated fungi. These organisms spend 7 decades growing underground, accumulating resources and expanding their networks in complete darkness. Then, with clockwork precision, they emerge simultaneously across vast geographic regions, fruiting in massive coordinated displays that can be seen from space. What triggers these septuagenarian emergences? The fungi appear to be counting something, measuring some environmental variable with extraordinary precision across multiple human generations. But what could they be tracking that requires such extended observation periods? Solar magnetic cycles? Gravitational wave patterns? Planetary alignments too subtle for our instruments to detect? The precision of the timing suggests that these fungi possess internal chronometers more accurate than our atomic clocks, capable of maintaining precise measurements across decades of environmental variation. Yet we have no idea what biological mechanisms could sustain such long-term timing systems, or what evolutionary advantages could justify waiting 70 years for a single reproductive opportunity. Even more perplexing is the discovery that some fungi can capture and utilize light despite lacking the basic cellular machinery that makes photosynthesis possible. These organisms possess no chlorophyll, no photosynthetic reaction centers, and no biochemical pathways for converting photons into chemical energy. Yet controlled experiments have demonstrated that they grow faster and stronger when exposed to specific wavelengths of light. The implications challenge fundamental principles of biochemistry. If these fungi can harvest light energy without photosynthesis, they must be utilizing physical processes that exist outside our current understanding of biology. Are they somehow converting photons directly into chemical energy through quantum mechanical effects? Are they using light to power biological processes that operate according to principles we haven't discovered? The phenomenon becomes even stranger when we examine the wavelength specificity of these responses. Different fungal species respond to different colors of light with remarkable precision, suggesting that they can discriminate between photons based on their energy content. This implies optical sensing capabilities that rival the sophisticated vision systems of animals, achieved by organisms that have no recognizable visual apparatus. But perhaps the most profound mysteries emerge from recent discoveries about fungal information processing and decision-making. In laboratories worldwide, researchers are documenting fungi that demonstrate memory, learning, and problem-solving abilities that should be impossible without neural tissue. Slime molds like Physarum polycephalum can navigate complex mazes, optimize transportation networks, and solve computational problems that challenge graduate students. But they accomplish these cognitive feats using cellular mechanisms that bear no resemblance to the neural networks we associate with intelligence. How does a single-celled organism store memories? How does it process information and make decisions without any centralized processing system? The questions become even more perplexing when we consider that these cognitive abilities appear to be distributed across the organism's entire cellular structure, suggesting a form of consciousness that exists at the molecular level. The discovery of fungal learning has forced scientists to reconsider the most basic definitions of intelligence and consciousness. If memory and decision-making can emerge from chemical gradients and cellular communication, then awareness might be a far more fundamental property of matter than we ever imagined. Indigenous peoples around the world have long spoken of mushroom teachers, fungi that can impart knowledge, wisdom, and insights that transcend ordinary human understanding. Western science has traditionally dismissed these reports as folklore or hallucination, but as our research reveals increasingly sophisticated forms of fungal intelligence, we're beginning to wonder whether these traditional accounts might describe real phenomena that we're only now developing the tools to study. The psychoactive compounds produced by psilocybin mushrooms create experiences that participants describe as encounters with non-human intelligences, entities that communicate through direct knowledge transfer rather than language, teaching concepts that seem to come from outside human consciousness entirely. While we can measure the neurochemical effects of these compounds, we have no scientific framework for evaluating whether the experiences represent genuine communication with fungal intelligence or simply projections of human psychology. But the consistency of reports across cultures and centuries suggests that something significant is occurring during these encounters. Participants frequently describe receiving information about biological processes, ecological relationships, and natural phenomena that proves accurate upon later scientific investigation. They report insights into the interconnectedness of living systems that align remarkably well with our emerging understanding of fungal network intelligence. Perhaps most intriguingly, many participants describe a sense of communication with a vast, ancient intelligence that views time and space from perspectives completely alien to human experience. This entity, consistently described across different cultures and individuals, appears to possess knowledge that spans geological timescales and encompasses the entire biosphere. Could these experiences represent actual contact with the distributed intelligence of fungal networks? Could the chemical keys that psilocybin provides unlock genuine communication channels with consciousness that exists at planetary scales? The questions push us to the very edges of scientific methodology, challenging us to develop new approaches to studying phenomena that resist conventional investigation. Meanwhile, researchers studying fungal networks in their natural environments continue to document behaviors that suggest awareness extending far beyond anything we can explain through current biological theory. Fungal networks appear to anticipate environmental changes, adjusting their resource allocation and growth patterns days or weeks before conditions actually change. These anticipatory responses cannot be explained by any known sensory mechanisms. The fungi seem to be detecting signals that remain completely invisible to our instruments, perhaps quantum fluctuations, gravitational wave patterns, or electromagnetic fields generated by celestial bodies. Their responses suggest a sensitivity to environmental variables that exist at the very boundaries of physical detection. The accumulated evidence points toward fungal capabilities that challenge our understanding of the possible. These organisms appear to process information through mechanisms that operate according to principles we haven't discovered, communicating through channels we cannot detect, and demonstrating awareness that extends across temporal and spatial scales that transcend individual organisms. In indigenous traditions worldwide, fungi are revered as bridges between worlds, organisms that connect the material and spiritual realms, the conscious and unconscious, the known and unknowable. Western science is beginning to discover that this metaphorical language might describe literal capabilities that we're only starting to comprehend. As you drift towards sleep tonight, consider that every breath you take contains millions of fungal spores. These microscopic emissaries carry within them the genetic memories of organisms that have witnessed the entire history of life on Earth. They remember the first emergence of complex cells, the colonization of land, the rise and fall of dinosaurs, the birth of flowers, and the recent appearance of human consciousness. Fungi comprise 25% of Earth's biomass, making them one of the most successful and widespread forms of life our planet has ever produced. Their networks extend through every ecosystem, connecting every forest, threading through every soil, linking the entire biosphere in communication webs that pulse with information we cannot decode. These ancient beings have been patiently weaving through soil and dreams for hundreds of millions of years, developing capabilities that challenge our most fundamental assumptions about the nature of life and consciousness. They represent forms of intelligence so alien, yet so essential, that understanding them might reveal what consciousness truly means. Perhaps the deepest mystery of all is why fungi remain so mysterious despite centuries of intensive study. With all our advanced instruments, sophisticated theories, and accumulated knowledge, we still cannot explain how they accomplish their most basic functions. The gap between our understanding and their capabilities seems to widen rather than narrow as our research progresses. This suggests that fungi operate according to principles that exist beyond the reach of our current scientific paradigms. They may be utilizing aspects of physics that we haven't discovered, processing information through quantum mechanical effects that we cannot measure, or demonstrating properties of consciousness that transcend our definitions of what it means to be aware. The questions that remain about fungi are not merely biological puzzles, they are invitations to expand our understanding of what life can be, what intelligence can become, and what consciousness might encompass. In their mysteries, we find reflections of the deepest questions about existence itself. As we continue to study these remarkable organisms, we may discover that the boundaries between life and non-life, between individual and collective consciousness, between matter and mind, are far more fluid than we ever imagined. Fungi may be showing us that the universe itself is more alive, more aware, and more interconnected than our current worldview allows us to perceive. The questions they pose may not have answers that fit within our existing frameworks of knowledge. Instead, they may require us to develop entirely new ways of thinking about intelligence, consciousness, and our place in the vast community of awareness that appears to extend throughout the natural world. In the end, the greatest mystery of fungi might not be what they are, but what they reveal about the nature of existence itself, and what they might teach us about questions we haven't yet learned to ask.